Hello everyone. So I'm Elif and I'm working in Man Savage's lab, and I'm here to present a software Angicart that we have been actively using it for almost more than 3 years. So in my presentation, I'll first give you an overview of our software, like its purpose, how the algorithm works, or like some advances on it. And then if I have a time, I will also give you some of the results that we have by combining the empirical data with a mathematical modeling perspective of the vascular system. Okay, let's start. So the purpose is getting a data from vascular images and the why. So we would like to extend our knowledge about the vascular system, like its structure, how it is developed or how it is evolved. And also that would allow us to test the current theories on the vascular system, like metabolic scaling theories or some reduced relationship between the parent and child vessels, which is called Murray's law. And also knowing more about the structure would also allow us to learn more about the blood flow. And finally, from a health perspective, so when you have a disease versus healthy tissue, the diseased tissue can also have some vascular abnormalities that also you can characterize them with such a software. And then it's also important for, once you know the structure, then you can also come up with some strategies for drug delivery to tumors, like diseased tissues too. So how it works, so Angicart accepts like 2D stacks of angiographic images. Then in those images, you see a lot of things that are vessels and that are not vessels. So in order to distinguish vessels from non-vessel objects, then you need to choose some intensity threshold. And then once you choose the intensity threshold, the algorithm does a skeletonization by using an erosion algorithm. Then at the end of the skeletonization, you have a big chunk of a vascular structure that is like a largely connected component, and which also has some center lines there. So then after the skeletonization, the software does the segmentation to get the whole topology of the vascular network and also get some physical properties of the vascularity, like some radius or length information of the vessels. So you, as an input, so you give some like angiographic images, 3D stacks of angiographic images, and you also provide an intensity threshold. Then at the end, the software gives you an output file describing all the segments. I can use that. How do you use it? Anyway, it doesn't. How do you use it? Oh, okay. So like for example, so for each vessel, we have a name that actually contains the endpoint coordinates and the length information, volume, radius, or number of downstream tips, or like the parent information, or some like ratios between the parent to child. So it's very actually useful, like it's very convenient to actually get it, like from images, you can get whole topology of the network using the software. And some frequently asked questions, is it free? Yes, but you just need to cite the paper that is published in PLOS Computational Biology in 2015. And so is it easy to use, install, and use? So the thing about this is that Angicart was written in a programming language called OCaml, which you may not all be familiar with. So, and also you need to use the command line to actually run the software. So if you're not familiar with like either OCaml or using a command line, then it could be a little tricky for you to actually get started using the software. But we have a website that I'm going to show you in the next slide that actually has some like directions for the installation or the usage. And also there's a recent work going on in translating Angicart in C++ that will also have some additional features in it. So it's going to be much more user-friendly in the future. And of course, it's always possible to get help. You just need to contact our group. So this is a website. So this is a very useful website actually for like, it has a link for downloading Angicart or like some installation details, guide and frequently asked questions, and it even have some example data and interpretation. And it also has some publication lists. So the next question is, what can you process via Angicart? So it can actually be applied to any like 3D image stacks. But so far we only have like an animal vascular system, we only analyze the animal vascular system. So this is a processed image via Angicart of mouse lung. But as long as you have like a 3D image stacks of anything like fungi network, neural network, or plant vascular network, then the Angicart would be able to analyze it or process it. But these actually fungi network or the neural networks can actually have some special features, like they can be loopy. And I actually forgot to mention, but it is an important detail that Angicart actually assumes that the vascular system is like a tree-like structure. But to be able to process these, like to characterize the loopy structures, the new version of Angicart in C++ is built such that it can also recognize the loops. So the advances, so recent work in translating Angicart to C++ also has some piece to identify the loops, which is especially important for like characterization of vascularity in plant leaves, neurons. Also, it's important for the stroke recovery because, yeah, the brain has some loopiness in it. Okay, so this is an actually early version of the Angicart in C++, and we tested it with the 3D toy network. And you can see that it's pretty easy to use. You just need to change the directory of the image, like directory file for the image, and it also has some nice visualization piece, so you can like rotate and zoom in, zoom out, and so on. So you can actually see the structure in detail. So this version can handle loops, and it also provides the topological data and the physical properties of each segment as Angicart does. Like the volume, the length, this is the name of the vessels, and so on. Like the adjacent like vessels also. And also, like in the future, we plan to have like to make it much more like user-friendly. We plan to have some GUI version that you can manually upload the images and choose a threshold, and then just push the button so that the analysis can be done very easily. Okay, so that was the end of the software like overview. And what we did with Angicart, so we have two different data sets, mouse lung and human head and torso, and these are the images processed through Angicart. With this empirical data, so we tested the symmetry assumption of the vascular networks, and in the literature, there's a general assumption that the siblings are identical in terms of their physical properties, like the radius, length, or branching angles. So we characterize the asymmetry by using some ratios between the radius, length, and branching angles, and we divide the smaller to the larger so that this ratio is in between 0 and 1. And so 1 means that siblings are identical, so it corresponds to perfect symmetry, and 0 may actually go towards like 0, it corresponds to perfect asymmetry. So when we plot the distribution, so for both like mouse lung and human head and torso, we discovered some schematic asymmetry patterns. So when we look at the radius and the branching angle ratios, we find that they are skewed towards symmetry, so they tend to be much more symmetric, especially compared to the blue one, the blue curve here, which is like a uniform, so they tend to be like much more asymmetric, and this holds for both the mouse lung and human head and torso network. And because we have these systematic patterns, then we ask the question how these systematic patterns emerge. And we have two different hypotheses. So the first one is related with the from an evolutionary perspective. So we hypothesize that selection and development influence the vascular geometry. So we have two different hypotheses for that. Maybe the minimization of the cost of the materials, or the distributing the flow efficiently leads to these branching geometries. And for these optimization like principles, we develop optimization functions locally and find a solution and compare with our empirical data to see if they actually match. And the other hypothesis is basically a random branching hypothesis. And for this one, we randomly place branching junctions by constraining the vascularity across different spatial scales so that it can go from like a local to global. So for the optimal branching hypothesis, when we plot similar distributions for the branching angles and the length asymmetry ratios, we find that actually the optimal branching hypothesis match the patterns at the network level with the empirical data. But we don't actually get a good match at the junction level comparison, which I didn't actually include the plot here. But this also says us that, and these are really local constraints, but this actually we concluded that a single or local constraint is not likely the only driving factor. So we need to actually maybe go beyond the local, or just maybe include like several constraints together to get a better match with the empirical data. So for the we also look at the random branching hypothesis. So the top row is schematic of our simulations that we actually have different constraints from local to global, and this row is the results for like simulations and the real network. And when we look at the global simulations, we find that it's really too bad at the predicting the real network. But when we actually look at the local or intermediate levels, they kind of like really match with the empirical data. We also did some statistical analysis between like these distributions and also for the distributions from the optimal branching hypothesis. And we find that actually what we find is really striking, that material cost optimizations are the strongest predictor of the vascular branching in the human head and torso. But for the mouse lung network, it doesn't really matter either we are looking at like local like random branching hypothesis or just material cost optimizations, because they're kind of like they give us a similar results. So it doesn't really matter. They give basically the same results, either local like optimizations or just local or intermediate random branchings. And this also leads us to ask more questions about the vascular system. So what is the real reason behind this result? Like, is it the tissue type, like head and torso versus the lung, or is it a vessel size because head and torso has larger vessels and mouse lung has like a smaller vessels, or is it between like a human versus lung? So, and really answering these questions really, and in order to better understand the vascular system, we really need high quality data. And Angicart offers an easy and efficient extraction of vascular data from angiographic images. And also we have a new version of Angicart in C++ that recognizes loops and will be much more user-friendly, and the full version will be available hopefully soon. And also, so I'd like to thank my group, especially Van, for all the support, and Mitchell for writing the software in the OCaml version, and David for his recent work on translating the software in C++, and also Christina Bostrom and Daniel Ennis for providing us data, and finally the funding resource.